From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 51, recorded on May 6, 2024. I'm Vincent Racaniello, and you're listening to the podcast about the nervous system. Joining me today from Salt Lake City, Jason Shepard.
Hey, Vincent, Aaron.
How you doing?
You know, we have a massive snowstorm up here, probably the biggest of the year. One of the biggest of the year, so a little unexpected. I think we have like 2 feet of snow up in my house right now.
Is that normal for snow to land?
We get snowstorms in May, but having this kind of big, massive one, unusual.
Huh.
Also joining us from New Orleans, Vivian Morrison.
Hey guys. It's hot here.
Warm,
it's going to be a heat wave. Yeah. So no snow.
And from New York, Tim Chung.
Hi everyone. I can't remember, there's a stat about every degree of warming you get in the atmosphere, it means that the air can hold X amount more water. So whenever it rains, it's going to go much, much worse. So that might be one of the reasons why.
Wet bulb temperatures.
Yeah.
That's bad news. Bad news bears for those of us who breathe air.
It's been a crazy tornado season in the plains, so that's also part of it.
All right, today it's Vivian's turn
to
educate us. Vivian, it's all yours.
So I'm feeling that there's a trend in some of the episodes we've done that are focusing on neurodevelopmental disorders. So I promise that at some point we will not do something on autism or ADHD or whatever, though they're very interesting topics. And I
really like these because I never studied because as a Parkinson's disease researcher, I'm going the opposite end. So it's good to have developmental. I mean, it's good to try to eradicate study developmental disorder.
Yeah. And I mean, this is new for me too, because actually I do cellular and molecular neuroscience, but I think I'm not sure how much people want to hear about. And then this protein bound to that protein. And, I think it's a little bit less inviting. But anyway, today we're going to be talking about a paper called The Developmental Timing of Spinal Touch Processing Alterations Predicts Behavioral Changes in Genetic Mouse Models of Autism Spectrum Disorders, plural. And the first author is Annika Tasnim, and this is out of David Ginty's lab at Harvard. And the almost senior author is Lauren Orefice, who is actually a graduate of the Georgetown program that I attended, and we were, doing science back to back. So it's kind of cool to see where she's gone. And so in a previous episode, I presented a paper about pain processing in a mouse model of ADHD. And so when I saw this, I thought this kind of intertwined with that. And this is about touch processing in mouse models, plural, of ASD, autism spectrum disorders. And just really quickly, autism spectrum disorders, I think it's important to say disorders, plural, because it comes in many different shapes and sizes, differing severities, and different systems in the nervous system are affected, and also just throughout the body. You can have syndromic disorders where there are multiple organ systems involved, and what makes autism research tricky is that we can't always tell which systems are involved, to what extent, how they contribute to the most debilitating aspects of these conditions. And so it's a little bit of a chicken and egg situation. So that can be tricky. And that's why it's so important to do the kind of work that they're doing here, where they're trying to actually bring up and figure out why different mouse models of ASD look different and respond differently to things. So the sensory processing differences is a robust feature of neurodevelopmental disorders and autism in particular.
So Vivian, I got to question. I don't know whether you know. I'll just ask anyway.
Yeah.
So in mice, I'm guessing different genetic models have different presentation of autism.
Yes.
How about in humans? Are there also different? Because I know like males and females present very differently in autism, and I think like historically females are usually underdiagnosed because they present so differently. So I'm just wondering, is it also like a wide range of symptoms in humans?
Yeah, it actually is. They mention in their introduction a couple of different well-known models, like the Shank3 knockout, and if you were to go look up, there are many papers. Actually, people are interested. You can go look up mouse models of autism, and you will find quite a bit of literature out there just reviewing the different kinds of models there are. Some of them might be the loss of a gene everywhere. Some of them might be a loss of a gene in a particular subtype of cell in the brain. And then you'll also get a sense of the different kinds of methods that are used to test these mice to see like to what extent they have symptoms that resemble those found in humans. I think the DSM criteria for autism is narrow. It's actually very limited, which means then that the individuals who could fall under that umbrella, there's a lot of them. It's like extremely restricted interests and communication difficulties or communication differences. But even within communication differences, for example, you can have individuals who like communication is very broad and diverse as a term, and so there are lots of components that could be disrupted while others are not. So there are some. There is an autism spectrum disorder where, for example, the individuals are hypersocial, so they have no boundaries. They don't understand stranger danger. But then on the other hand,
mice. The genetic models of mice, there are sex differences as well in the behavioral outcomes, and even some of them, are X-linked. So Rett syndrome, which is a common intellectual disability, autism spectrum disorder in humans, it's well modeled in mice, and that's where it's also actually more prevalent, or it affects females more. And that's partly because the males, where that mutation is actually not viable, or it's very unusual to even have that mutation in a male survive. So but I would say, the intellectual disability genetic mutations are quite well known and modeled in mice. But the general autism, spectrum disorders that are not associated with a specific mutation, that's where it gets really difficult to model in mice. And I think partly it's, as Vivian said, the diagnosis, it's based on symptoms, it's based on symptomology, and then there's sort of this broad category. If you have 3 or 4 of these broad category deficits, then you have what's called an autism disorder. And that's, I also think, partly why the number of children that are diagnosed has gone up.
Yeah, which is interesting. I hear so many people who have, who are on the spectrum, and I start to wonder, well, aren't we all on the spectrum in differing ways? And anyway, but I think, Jason mentions the word diagnosis, and I think something that, putting all this variability and heterogeneity aside, something that we all can agree on is that it would be best for everyone involved if we could detect and diagnose autism and get a sense of maybe the severity or what's coming, develop some sort of prognostic idea as early as possible. And so sensory processing, spinal touch, is not something that necessarily involves or it's something that we can detect or that we can test very early on, which is what they're going to do in this paper. And so then that could presumably lead to earlier detection and diagnosis, and then that could probably help us put into place therapeutic measures as soon as possible, because we do know that, this is about brain plasticity and brain connectivity. And that stuff is not set in stone. I mean, it's plastic. So we do have ways to address this. And they even say that it's possible that there are therapies that address sensory processing specifically that may then have like spillover effects on other aspects of the ASD symptomology. And so something that this paper brings up is a concept that was actually coined or introduced by in a 2017 paper by Georgiadis, Bishop, and Fraser. And the concept is chronogeneity. And so I'm going to read a quote from the paper that we're presenting today. Chronogeneity describes diverse clinical trajectories that individuals with ASD can take throughout development and onward. And it posits that temporal and cross-sectional heterogeneity across individuals may represent a form of informative variance that describes a more dynamic and well-powered understanding of ASDs. And I like that because it's not shying away from the fact that this is hard and it's complicated. And I think that speaks to the kind of purpose of this paper and this general line of work. And so here they're looking at chronogeneity in these mouse models, and they present evidence that one noteworthy aspect of chronogeneity with real-world relevance is the maturation of circuits and circuit components that regulate incoming touch information. So I want to just again, this was new to me, or like it was like way back in grad school I learned about this stuff. But I want to review quickly the circuit that we'll be working on because the kind of location of certain things will be important.
Actually, Vivian, before you do that,
sure.
I just want to throw out some numbers because I was surprised by this because when you said we're going to look at autism, I just assume autism is largely a social kind of social symptom and also communication symptom and maybe restricted interest. But in the first line of the paper it says that sensory processing dysfunction, so just how you interact with the world in terms of touch and also maybe visual input, it affects 94%.
And auditory.
And auditory.
And gustatory and olfactory, pretty much all of it.
So is it all? So it's non-mode dependent. It doesn't matter which.
It varies though.
Yeah.
It's not like someone will have all of them.
Okay.
But at least one of them.
But it affects 94% of people
Yeah.
with autism spectrum disorder. So
That actually makes a lot of sense when you think about it, since your nervous system is all about taking stuff in and then crafting the appropriate response. And if the information coming in does not reflect what, reality really, or it's going to have different outcomes.
Yeah, I mean that's also why a lot of the social interactions are confounded by the fact that they have a problem with just sensory processing. So being able to, when they go out into the world, there are so many different things coming in, and the brain is finding it difficult to make sense of it. So that's why they like, very simple environments. They're saying that's why the restricted interests are partly also due to that. So I actually think, the sensory deficits are one of the main reasons why people have those cognitive effects.
That's interesting.
Completely agree. And, Tim, your comment makes me realize that I take for granted that people know this because, I have ADHD and that overlaps a lot with ASD. And so I do have, and I experience hypersensitivity to light, sound, and touch. And I don't know if I talked about this when I talked about ADHD or the ADHD pain model, but that soft touch, what is supposed to be pleasant, can be interpreted as painful. So I couldn't agree more with what Jason said. And thank you for bringing it up because I tend to overlook the fact that most people might not think about ASD like that. So the touch circuit. So you have in your skin, you have receptors that are sensitive to pressure, among other things. And that's going to send a signal to what's called the primary sensory afferent or primary sensory neuron. And its cell body actually resides in this little ball that we find outside, but right next to the spinal cord. And this is called the dorsal root ganglion. And so it's receiving input from the outside, and then it has its cell body, and then it projects its axon into the spinal cord. So only the axon is in the spinal cord. Then there's a series of relays that happen inside the spinal cord involving inhibitory interneurons, as well as that are resident to the spinal cord, so they have their cell body and their axons and dendrites there, as well as descending terminals from neurons that are higher up and eventually connect to the brain. And then, and that's where there's a series of kind of feedback loops and mechanisms to kind of regulate what kind of output is stimulated by the sensory input, the sensory information coming in. And all of that is going to finally land on the dendrites of a motor output neuron in the ventral horn of the, well, I won't go into dorsal ventral horn yet, but a motor neuron that then is going to join the primary sensory afferent inside the spinal nerves, inside of your nerves, and go back out to your muscles to then cause you to, for example, like scratch your face or move away from something or move towards something if it's pleasurable. So the most important thing in all of that was just that for the primary sensory afferents, only their axon is in the spinal cord. And then we have dorsal spinal neurons that have their dendrites and somas dendrites and somas and axons in the spinal cord. And then the motor efferents have just their dendrites and their cell bodies, but the axon is outside in the peripheral nervous system. And there's a general theme in neuroscience, which I find really interesting, and that is inhibition. And I talked about how there's these, this mess of inhibitory interneurons in the spinal cord and then descending inhibitory inputs. And an example of this is a little morbid, so if you don't, plug your ears if you don't want to hear me talk about what happens when you are collecting early postnatal brains for doing cell culture or something like that. In order to collect the tissue, we need to decapitate the animal. And as soon as you decapitate the animal, the rest of the body starts moving a lot. And that's because the descending inhibitory inputs, in part, it's because these descending inhibitory inputs from the brain have been severed. So I think that's a good example of how inhibition is so important.
Is that why drunk people wander around so much?
I don't know.
I'm going to say yes.
I'm like, what made you think of that? Oh, yeah, okay, I mean, maybe. So like whether you're talking about drunk people or like mood or behavior or autonomic function or sensory motor function, inhibition is what we see or what we don't see, and others, what we experience or don't experience, has a lot to do with inhibitory control. And in this paper, they mentioned, so I learned from this paper, because I don't think about this stuff a lot, is that there's a couple of different ways to break down inhibition or inhibitory mechanisms. One is by the neurotransmitter that's involved. So we have two major inhibitory neurotransmitters, or two neurotransmitters, GABA and glycine. And it's well known that the expression of GABA or glycine receptors changes as a function of age. And I believe, among other things, it can change. But so this is meaningful that, GABA and glycine can be used differently in different times in different places. And then another way you can break down inhibition, kind of understand what it means, is has to do with the nature of the connections between neurons. And this is important for this paper, and it relates back to my, really hammering the point home that, the axons are going to be in the spinal cord, but not the cell body. And then here, the dendrites in the cell body are going to be in the spinal cord, but not the axons. Because they break it down to two kinds of inhibition: axo-axonic and axodendritic or axosomatic. And in the first case, axonic, you have neuron A projecting and synapsing onto the soma or dendrites of neuron B. And then independently, neuron C is synapsing onto the axon that connected those first two neurons. And so it's giving this, the neuron C is modulating the activation of B through A. I wish Tim could, he's so much better explaining this stuff in like, easy to understand language.
It's similar to like me and Vivian could be having a conversation, and I'll be talking to Vivian, and then Vincent comes in and say, "Tim, please shut up. The podcast is starting." So I'll be the axon that is talking to Vivian, and Vincent is here as an axon himself to shut me up so that something else can happen.
Right. That's a fantastic analogy.
But I think that the key aspect there is, as you said, the inhibition and sort of the signage of like how these things are modulating. The actual connectivity may not be that important for folks.
Right. And then the axodendritic, axosomatic is just that it doesn't involve this third party. It's just you have a neuron projecting directly onto whatever the output neuron is. And in that analogy,
Vincent would be telling Vivian to stop listening to Tim, just to complete the.
Thank you. Analogy. No, that's fantastic. I was just at an elementary school today talking to third and fourth graders about the brain, and we were talking about, I encourage them to come up with analogies like that. So I think that whether you're a third or fourth grader or,
I'm a fifth grader by now.
Or much older, those analogies are really helpful. So I'm actually going to present the story in a slightly different direction than the authors presented it, because I felt like they led with the really interesting stuff, and then for the rest of the paper I was like, meh. But since we're talking about like spatial and temporal restriction of different kinds of inhibition, we're going to start at the end of the paper. And I am sensitive to what Jason was saying, that we don't, and part of this is because I just want to get through some of this, like, oh, this is expressed here, and this thing gets turned on by this and that, but not the other thing, because that's a little bit more difficult for people to like put into a context and like feel, is useful. But for the people who are a little bit, who are more interested in this kind of like mechanistic stuff, I'll mention it. Several of the mouse models of ASD that we have involve the genetic deletion or loss of function mutations of elements of GABAergic and glycinergic neurotransmission. So in this paper, I'm just going to highlight two of the mouse models that they use. One is the GABARB3 or GABA A receptor beta 3 subunit mutant. And then the other is the NLGN2 or neuroligin 2 mutant. And both of these mutants show tactile hypersensitivity. And we'll talk a little bit more about some of the other behavioral tests that they do, where we really start to see these two models diverge. But the GABARB3 is part of the GABA receptor complex, which is made up of a couple of subunits. And then neuroligin 2 is involved, at least in the brain, in the assembly and the kind of localization of GABAergic receptors. So what they're actually, but I believe at least at the time that this was published, I think the function of neuroligin 2 in the spinal circuits was not known. So what they find actually is that,
generally speaking, neuroligin 2 or neuroligins are those sort of cell adhesion molecules that help build synapses, and also sort of the way they build synapses, that they can bring the pre and post synaptic compartments together. Mm-hmm. And then in some cases, sort of like neuroligin 2 seems to be more important for inhibitory synapses than for excitatory.
Right. And so I think what's interesting is that they actually find that in this particular context, neuroligin 2 doesn't have anything to do with organizing at least GABAergic synapses, which I think is potentially a new piece of information. But again, it's not my field, so I'm not sure about that. But both of them affect tactile sensitivity in life, but it turns out that the GABARB3 based mechanism is something that takes place early in development, whereas neuroligin 2, that mechanism doesn't really come on until young adulthood. And so then they use a series of genetic tools that I'm not going to go into, but it's based on the Cre-Lox system, which I think that we've talked about before, where basically you can specifically knock out a gene in one cell type or a class in a more restricted way. So you're not knocking out it everywhere. And they use these genetic tools to locate to figure out where GABARB3 and neuroligin 2 are expressed. And this is where they figure out that, GABARB3, so the thing that's that's necessary for these GABAergic receptors, is expressed on the primary sensory neuron, so the thing that's coming in. And so since it's only the axon that's in the spinal cord, so that means that these GABA receptors are not where we would typically think of them as being, the postsynaptic bouton, but they're actually on the axon. So we're talking about axo-axonic or presynaptic feedback inhibition. And that they will show that it comes on very early in development. And then neuroligin 2, they find out, is expressed in dorsal spinal neurons. So they're entirely contained within the spinal cord. So we've got these temporally and spatially restricted inhibitory receptor components. And they're not, so I mentioned already that one is, that neuroligin 2, it turns out, is not going to it doesn't really use GABA, it's glycine sensitive, and the other is GABAergic, it's GABA sensitive. So I won't go too deeply into that other than to say that they use some cool ex vivo preparations. So they actually pluck out a dorsal root ganglion, which contains those primary sensory afferents. In another case, and so they'll use that to look at GABARB3 function. And that's where they show that if they can drop some GABA on those neurons, they can generate, or on that prep, they can generate this, in these inhibitory currents. But then if you do the same thing with a dorsal root ganglion derived from a knockout animal that doesn't have GABARB3, then you get nothing. So again, it's just kind of really honing in on that, like this is where it's happening. And then they do a similar thing for neuroligin 2, using a spinal cord ex vivo prep. And so, like I said, we don't need to go too deeply into that, but basically they find out that while there is some activity that this neuroligin 2 complex that it's associated with, it's receiving some information even early in development, but it's really weak and infrequent, and it's not GABA based. But as the animal ages, it's receiving much more glycinergic input, and that's where we really see this axosomatic or axodendritic inhibition coming into play. So far, they've shown us that these two proteins that we already know from previous literature are ASD models, are working in different cell types, and they're working at different times because one is showing up very early in development and another doesn't show up until later in life. And they're working through different neurotransmitters, but they're both providing input in different ways to spinal touch process, or the touch processing. And so having described this spatiotemporal segregation of these different mechanisms, they want to see what the behavior is like, what happens to the animals. Yeah, sure.
Before we go there, so just to recap, in very early mouse, so like these are P4, like postnatal day 4, so these are mouse pups that are still like pink and hairless. They found that
hairless, sort of.
Okay, maybe
because
slightly
that's a
hairy.
big component of what's coming up next. is
Oh,
that
okay,
they
sorry, my
do
bad,
have
my bad.
hair.
Yeah, okay, but like very early pups.
Very early.
Very early pups, the kind of periphery sensory neurons, they can actually get inhibited by GABA. And then as the mouse ages, there's not extra inhibition that is available. So these neurons almost kind of mature quite early already. Whereas inside the spinal cord, where there's a lot of so-called feed-forward inhibition onto the postsynaptic cells, those actually don't have very strong inhibition early on during this early postnatal day. But as the mouse ages, there's much more inhibition, and you get and most of it is by glycine. So the question is, what is the function of the peripheral sensory neurons that makes it necessary to mature early, and what is going on later, such that inside the spinal cord you can wait until a later day before it matures, or maybe that's on purpose.
Yeah, I don't know, but I do think that's a really interesting question. Not one that they go into at all in this paper, but I don't know if Jason, you have any thoughts about that. I do not.
Because it seems like
but
if
it
this
is an interesting kind of
if this is messed
separation.
up, it is potentially linked to autism. This is exactly what they are going to show behaviorally.
Right.
Yeah,
But then again, why would the early inhibition be necessary, but not the later one?
Yeah. Anyway, I don't know what the function of this critical inhibition is, but it seems like it is
I
very
don't know as much about the sort of peripheral inhibition and how that works. I mean, in the central nervous system, we know that the inhibition develops much later than initially the excitatory transmission, and some of this is due to try to keep that brain plasticity optimal in the beginning. And I don't know if that's the case for peripheral wiring, and that you sort of have a lot of plasticity going on. There's a lot of wiring going on. And, I think that's another common theme in autism, is that these developmental windows of plasticity are a bit disrupted early. And that's why it's a developmental disorder, because you have these abnormal amounts of plasticity, either too much, too little. And that's why you get this wiring defect.
Yeah, absolutely. And I think, and I'm going to bring this up later, but I'll say it now, that the big whoa, this is really cool moment for me came when they were talking about how they used the different genetic mechanisms to knock out, or genetic modifications to knock out GABARB3, for example, only in the primary sensory neurons. So everywhere else in the nervous system is still making this protein. But by knocking it out just in that cell type, that is sufficient to generate social deficits and anxiety behaviors in adult animals, which I thought was fascinating, because then it just highlights how important, and it's I guess just again pointing back to your question, your comment, Tim, it points to how important and why, who knows, incoming sensory information in those loops are to the development of stuff up here that's that we see as being much higher order.
Yeah, I always assume it's
complex.
Deep in the brain, something goes wrong when you have autism, but no, it's actually outside in your peripheral nerves.
Which I think speaks also to what Jason said earlier about how that sensory processing differences is probably one of the if you were to really get down to the heart of it, that is probably a big factor in the development of this range of disorders. So let's talk about the behavioral consequences of all of this. So they're going to look at tactile sensitivity and sensory motor gating, anxiety-like behaviors, and social behaviors. And for this section, these are all features of ASDs, and there are things like I mentioned earlier. We have the batteries of tests where we can look at these kinds of things and kind of then be able to place the different mouse models kind of in groups and be like, okay, well, these ones show anxiety-like behaviors or repetitive behaviors like excessive grooming, and these ones do not. And then we can use those differences to try to figure out why. And then hopefully that will reflect back on what the kind of heterogeneity that we see in humans. And I want to emphasize here that they are looking just at adults for this section. So how do they measure tactile sensitivity? So they do two things, but for the adults, they do this thing called a pre-pulse inhibition assay. And they're going to do a tactile version of this. So the standard acoustic or like hearing-based pre-pulse inhibition assay, or PPI, has an acoustic startle stimulus, is something really loud that makes the mouse freeze, and you measure the amount of time that the animal stays frozen. And then in subsequent trials, you introduce a pre-pulse, which is not at the threshold that's going to stimulate freezing. You introduce a pre-pulse, and then you slam them with that freeze-inducing stimulus. But the presence of the pre-pulse, that little like kind of signal that something is coming, actually will reduce the amount of time that the animal stays frozen. And so if we see that kind of reflex modification, that's an indication that we have normal sensory motor gating. We have these inhibitory circuits to help modify behavior. And then the tactile version of this is that instead of having an acoustic pre-pulse, they have a tactile pre-pulse, which is just a puff of air. And so I think that's pretty cool.
It's literally a jump scare.
Yeah. They jump, and then you're saying that are they measuring like how high, or is it time, freezing time?
Well, and this is similar to, so you can induce the same thing in humans. This is kind of why this is sort of, I think we may have talked about PPI before and schizophrenia. It's sort of this weird telltale of schizophrenic cognitive dysfunction, that they have deficits in this PPI. And so it is something that you can observe in humans and in animals or in mice. Yeah, and it's fun for those of us who, do have sensory processing differences to wonder.
But it's interesting. It's interesting.
We don't have that.
It's interesting that in this version, so in the classical pre-pulse inhibition, you play a quieter tone and then slam them with a very loud tone to really make them startle. In this version, instead of the quieter tone, you just touch them quite lightly. So it's a tactile pre-pulse, and then you hit them with a startle tone. But that still works. So it's cross-modal.
Yeah.
So
no, that's really
quite
interesting.
I didn't expect that to work, but apparently it still works. So that's quite interesting.
Yeah. Yeah, that is really interesting. I didn't think about that, but it's true. So basically they showed that they're mutant mice. They're GABARB3 mutants and Neuroligin 2 mutants as adults. Because they're, as we know now, that they're in the inhibitory circuits are not working properly, they both will show this freezing. Wait, is it just that, or?
No, the pre-pulse has a stronger effect. So they're more sensitive to the pre-pulse.
Yeah. But the reason I paused is because I was so there is a divergence of these two models, and it doesn't have to do with the sensitivity or the tactile sensitivity or sensory motor gating. Both of these mutants are affected. But where it does diverge is when we start talking about anxiety and social behavior. So I just wanted to check myself there for a minute to make sure I wasn't saying the wrong thing. So like I just mentioned, then we start to see a divergence. So they have...
But maybe it's important to notice that these are tested in adult mice. These are...
Oh, yes, I mentioned that before. Mice, yeah. But bears repeating. These are adults. So as adults, these mutants both demonstrate this hypersensitivity to touch and reduce sensory motor gating. So one of the aspects that are, one of the behaviors that's commonly measured in a lot of models of a lot of things in neuroscience is anxiety. And we probably talked about the open field test before as a way to measure anxiety, as well as locomotion in general, and other like exploratory behaviors. But basically, it's just a box or a circle. I don't know. I'm sure somebody has feelings about whether it's a box or a circle or a trapezoid or whatever. But there's an inner zone, where, they're just kind of wandering around. There's no walls close to them. And then there's an outer zone where the mouse gets to be close to the wall. And mice are exploratory by nature. And so they'll wander around in the inner zone, the kind of open part. But then if an animal has like a reduced exploratory drive or increased anxiety-like behavior, they'll spend much less time in the inner zone and kind of hang out in the outer zone next to the wall as much as they can. And so in their studies, they show that under normal circumstances...
That's a natural behavior because they're prey animals, and so you don't want to be in the open where some bird's going to come get you, right?
Yeah,
So in the
and
wild,
then it goes the other way too, right? There are some models where they'll spend just a lot more time in the inner zone wandering around, which I think I can imagine might be related to their ability to, their risk tolerance or something like that. Or the circuits that are involved in not getting eaten, for example.
As someone who's had plenty of mouse infestation in my apartment, don't know whether I should own this up into public. My mice definitely hug the wall when they come out. But then if you walk on the street, those New York mice definitely don't really care about the wall. They just come and
They're
grab
mice? They're not rats?
Also rats. They fight it out.
They fight it out. That's funny.
I'm sure the New York rats or mice are like a unique evolved breed at this point.
We need Vincent to come and genotype all these mice that you get on the street to see how much they've diverged.
That's a really good point. And something to keep in mind about mice is that the mice in here are not the same as the mice out there.
Right.
So anyway, they find that that in their mutants, like we said, they're both, as adults, they're both hypersensitive. But then the neuroligin 2 animals, so they are missing that kind of the inhibitory mechanism that comes on later in life. They perform just like the controls. They explore the inner zone for about 40% of their time, and then 60% of their time is spent in the outer zone. But then the GABARB3 mutants who are missing this early inhibitory mechanism spend 80% of their time next to the wall. So that suggests that there's increased anxiety. And then they go on to also test sociability. Because as we talked about before, anxiety I don't think always shows, I don't know if anxiety always shows up in autism, but I think if you have social difficulties, that could cause some anxiety, but I think, I don't know. I don't think it's included in the criteria, diagnostic criteria, but I would say it's probably pretty common. But sociability and social difficulties are pretty common in ASD. And so here they use something called a 3-chamber test. And there are actually a couple different phases of this, so you can test a couple of different things, but the phase that they're using is a novel mouse, the option to explore and say hello to a novel mouse, or to explore, in this case, just an empty cup. And sometimes you can have an object inside the cup as well, but in this case, it's just the empty cup. And then you report the fraction of the total trial time that was spent in one chamber or the other. And under normal circumstances, the control mice will prefer to spend time with, a mouse rather than an empty cup. And so interestingly, they show that their GABARB3 mutants make no distinction between the mouse and the object. In fact, I have to remind myself of the graphs because I don't have them in front of me, but I think I stopped and asked myself whether they actually spent more time with the object, the empty pencil cup, than this mouse.
Noble mouse. It's pretty insulting if you're the mouse, sitting there and trying to strike up a conversation with this other mouse, and they literally run away from you and go interact with an inanimate object.
Yeah. But so I've been working on this 3-chamber analysis with my mice here, and I have found that time in the chamber does not necessarily reflect what the animal is doing. So I think if you see a difference, great. You could arguably stop there. But if you don't see a difference, like if you don't see behavior that is, if you see the animal performing in the same way as your controls in terms of how long they spend in one chamber versus the other, they can be in that chamber, but be like staring at the wall or like reorganizing their poops or something, like, which I have seen them do. They'll like pick them up and like move them in corners, like stick them in little holes in the wall, stick them on the wall. So it just is not capturing what the animal is doing. And like, I don't know why it would be in one part of, in that chamber, but like not be interacting. But I think it's just an example of how like, you can always go a little bit deeper, and we don't actually know, like, is this pleasurable for the mouse? Is it like
there has been a lot of criticism of this
aggressive?
3-chamber assay for mice, partly because of this, and kind of like develop better social, social. I mean, because mice are definitely social creatures, but these kinds of sort of forced assays, some of it's just hard to interpret. But
Yeah. But I think I've seen a couple papers recently that are using like AI to like catalog everything that the mice are doing. So maybe from that, we'll be able to say, like, this method can still be used, but you have to analyze it more deeply. You cannot assume that just because it's in the chamber, it's because like if you look at the figure of the paper, they actually have a heat map that shows like the different colors indicate how much time they spent in a particular area. And so for the control mice, you'll see like the mouse cup and the empty cup. And for the control mice, there's like a bright yellow circle around the mouse cup, but not as much around the pencil cup, the empty cup. But I've also seen in my own work that you can have the mice sitting and ambulating right around the cup that has the mouse or the object in it, and they don't turn and interact. It's like they just kind of orbit the cup, but then they're not actually like poking their nose in or directing their, So I don't know what that means, but I think just it's another example of how you can take these analyses even further because we don't really know what the mouse is experiencing.
yeah, and of course, there's compounds here, and like, okay, so you've got a novel object, but then if you've got sensory deficits, can you tell the difference between the novel object? Does it even cue the animal versus there's this high anxiety, so maybe they don't want to interact with another conspecific. They'd rather interact with the novel object. So there's all these different ways of sort of interpreting that data.
Yeah, but I think what's nice about the anxiety test and the 3-chamber test, both of them have their caveats, as all of these methods do, is that they help differentiate these two mutant models, which they have shown are spatially and temporally restricted. So I think that's cool. So then we're going to go to figure 1, and we'll finish up this paper. But so we looked at. We identified where these proteins are and how they're acting and when they're acting. And we've seen that even though as adults, both mouse models have tactile hypersensitivity and altered sensory motor gating, it's really when you get to like the anxiety and the social skills tests that you see them diverge. And so the neuroligin 2 animals don't have any of these. They don't have this anxiety-like behavior, and they show normal social preferences. But these are adults. And we know that the GABARB3 based mechanism is showing up at very early on. And so they say, how early does this turn on? And so they look at these P4 animals, postnatal day 4 animals, and they have to non-invasively look at sensitivity. So what they do is they have this little platform, and they put the mouse on it. I think they like stick. I read the methods, but I don't quite remember if it's. I think they may have stuck little dots. Is it on the mice, or I
saw the video.
Them?
I think it's like computationally generated.
Okay, so because I was like, that's a caveat because they can feel the stickers. But
Yeah,
they place
just
dots on it, and then as the animal moves, they can say, well, how many of those pixels are now obscured or have moved.
changes. Yeah, they do like a flow
Yeah.
map.
Yeah. And the hypothesis is that probably their GABARB3 mutants will show this very early hypersensitivity, and then that leads into what the title of the paper is, right? That could be predictive of later anxiety and social deficits. So they have like this little platform, and they will then do a little puff of air on the animal, and this will cause the animal to move its body, displace itself. And they do a couple of different versions of this. And they do it on the back where there is a little bit of hair growing, not much, but a little bit, enough to create what they need. And so they measure how big the body displacement is of the animal after the puff has been given. And they also test whether the animal become habituated to the puffs, if they just like repeatedly are puffing the animal. So the control animals will consistently move, with increasing intensities, as do the mutants, but the mutants move a lot more, the GABAR3B mutants. And they don't habituate. So they will continue to have these large body movements and displacements, even after repeated air puffs are applied. And so this is in line with what we saw about when these different inhibitory mechanisms come online.
So this actually answers my previous question about what is the purpose of these kind of inhibition to sensory neuron. Is one of them might be for habituation, so that you don't keep, if you're a mouse
in a bottom of a pile of pups.
Yeah, exactly. Or like if you're a mouse and you're sitting on bedding, which is like sharp edges of like sawdust or whatever, you might not want to keep feeling that sharp corner if you're sitting
and it's kind of like with our clothes, right? Like we don't, you might put it on and be like, oh, that's a little itchy or whatever, but then after a while you stop, you become like kind of blind to it, so to speak.
And the hypothesis that is the GABA that is shutting it down. Yeah.
Mm-hmm.
So Vivian, why in this displacement graph it goes up and down and up and down? Like they get habituated? Is that it?
You mean the GABARB3 mutants? How it goes like this?
Yeah,
I don't know, but I thought the same thing. Does anyone did it? I don't know.
Yeah, I saw
it does after like. Yeah, it's like up and then down.
In panel
I don't know if it's...
you can tell it actually does habituate, because like
Must.
During the first 4 trials there's a decline, but then afterwards it goes like wildly like up and down. So it seems almost like the mouse has 2 modes, and every now and then it would go into this mode where it like strongly reacts to a stimulation.
Yeah, and I think it would be interesting to look electrophysiologically at that. I mean, I'm sure that would just be practically challenging, but
yeah, I'm guessing
the
right
timing
now
is interesting.
We don't know why it goes up and down. Yeah.
Mm-hmm. But I don't know. But I also noticed that, and I was like, "Huh, that's interesting." And that was just a representative trace. Representative, and I know that scientists are not always the best at choosing a representative trace, but that one, it's not like it just fits seamlessly with, because you had the question. We all had the question, like, well, why does it go up and down? So I'm tempted to say that probably was, representative of the kind of the individual variance that they're seeing.
But they would argue that at every point is higher than the wild type. So
that's what counts.
Even if it goes up and down, even in the first 3 or 4 where it does start to habituate, they're higher up.
It's still higher. They're still hypersensitive to tactile stimulus. Yeah, for sure.
Yeah. So then the last experiment, which was actually like one of the first experiments in their paper, but the last experiments in the way that I'm choosing to tell the story, is they're like, okay, P4, we see that this hypersensitivity is already in place. Can we go even earlier? So they use, and this was cool. I was like, whoa. They use optogenetics. So they've genetically modified the animal so that only a particular kind of cell, this low threshold mechanoreceptor, these terminals, will be activated by light. So it's a way to essentially create that like touch experience without actually touching the animal. And they do this in P0, so like the day the animals are born, and they also do it at embryonic day 18.5, which I was like, whoa, that's cool. But basically, they're able to, stimulating these neurons using light, they're able to generate the like motor responses in these GABARB3 mutants, similar to what they saw in the postnatal day 4 animals. So this suggests that this hypersensitivity is happening in utero in these mutant mice, and that in utero we already are having that GABA, axoaxonic or presynaptic feedback inhibition loop coming online. And I was also thinking, like Tim was mentioning, the bedding, the kind of like mulch-like bedding that the pups have, or that the mice have in their cages. And I was thinking, they're constantly being touched by like the sac that they're in, and in mice they have a horned uterus, and they can sometimes have litters that are like 9 or 10 mice. So it's a lot of mice packed in there, and so I don't know if there's like a reason necessarily, but if they're, it's already occurring very early on when there's probably a lot of incoming tactile sensory information. So I was kind of thinking, well, like, what do we do with that information? And because we were at the very beginning of this episode, I said, the hope is that with the more we learn about this stuff, the more we can figure out how we might be able to do early detection, early diagnosis, and then early therapy. And I thought, is there a way to do this in utero? Like, we do genetic testing to see, I think the kind of like basal level of genetic testing is to look for chromosomal abnormalities. But in individuals who are, there is a history of autism, I think you can go ahead and have these genetic tests done to see if the child has a mutation in some of the genes that have been identified as being causative. But then I thought, well, what if you also have this thing that you could somehow measure, I don't know, in utero, and what if it could help predict the severity of symptoms or the kinds of symptoms that the individual will come to face, outside the womb. The authors are not saying that.
Yeah, I would say that's
they are not saying that, but I am just musing, as us basic scientists want to do, about how this is useful or how it could be useful or not.
It actually makes sense, because I think back in the day when people were perhaps underdiagnosing female autism patients, there was at least if I remember correctly, there was a hypothesis that the reason why there's so many male autistic people is because in utero you are exposed to too much male hormones. And that ends up causing your brain to be like more of a male phenotype, which is people who are very antisocial and like.
No, that's so rude. That's like not true.
Probably, but that's like research done like maybe 20 years ago.
Yeah, I wouldn't say that's the conclusion.
Like,
we should be.
But I think like people speculate whether there are like exposure in utero that might be causing things to diverge.
Yeah, and it's like what we were talking about, oh my gosh, what is it called? Was it the last episode? We were talking about COVID. And
that was the last episode, yeah.
Yeah, that was the last episode.
Jason's.
And I asked, what about in utero? Because I think the protein that that COVID was interacting with is actually an identified like cell adhesion molecule or something that's important for directing axon targeting at least in the hippocampus. And so I think, there's something to be said for what is happening prior to birth, since so much neurodevelopment is taking place then and kind of laying down the foundation for the nervous system. So I think that that's good questions to think about and kind of like, is there anything one can do?
Issues here, even in this study, they show this, right? That even genetic forms of autism have different. they may have these tactile deficits, but then they show different behavioral symptoms. And I think we have to be really careful about saying, okay, well, if we measure tactile dysfunction really early, you can't tell the parents, oh, well, then they're going to get X. And I think there would have to be a lot more sort of studies to figure out how diagnostic it is.
Yeah.
Although one of the paper, so I went back to look at some of the previous papers from this group, and the previous paper from Lauren Orefice in 2019 was actually absolutely fascinating because you can knock out these genes that are autism related, and by modifying the Cree a little bit, you can knock out these genes at different developmental time points. And she found out that a gene that if you knock out globally, throughout your entire life, that would cause both tactile and social deficits. If you knock the same gene out later, like around like P28, and then test the mouse in adult, it would still have tactile deficit because you knocked that gene out, but the social deficit is much reduced or gone, and the anxiety. And conversely, you can switch on you can rescue. So in a mouse that has that gene knocked out during development, you can rescue it in adult. And she found that if you rescue it, it doesn't work if you rescue it in adult. Like by the time you have grown up and you're trying to put the gene back, you might be able to rescue the tactile hypersensitivity, but the social deficit and the anxiety didn't get
already
rescued.
in place.
So she suggested there's a critical window during early development where these things are very important.
Right.
So in that case, you might really have to act fast if you want to. There might be a limited time. So,
yeah, I know. I mean, I think this is a big issue, even with treating these genetic forms of the disease. When you know the person's going to have the syndrome, so Fragile X syndrome, Rett syndrome, these are, now very commonly first diagnosed at birth with a common, sequencing or genetic tests. And even those trials where Fragile X syndrome, they had a trial, a couple of trials trying to target some of the pathways, and they found that some patients did actually respond, and most others didn't. And part of that was because they were only allowed to test teenagers or more adult patients because of the IRB. So I think that's going to be key. I think even in mice, so some of these mice, you can rescue the final some of the symptoms as an adult, but not all of them. So
yeah, and I think anybody who's spent time with children sees how everybody's developing and reaches different milestones at different times, even people who are considered like typically developing. So I think that just further complicates it. And to Jason's point, you can't rely on any one thing at any one time to point you in the right direction. And I mean, we need a lot of data points on a lot of different things for a longer period of time. And so if something comes out of this down the road, then it should that could be applicable clinically, then it should just be, one tab in a very thick binder of things that we need to look at and track in individuals. And then, what would be fantastic down the road is if we can draw a link between what we've learned there from development and collecting data over long periods of time in many different modalities and many different people, and connecting that to therapeutic outcomes. I mean, that would be fantastic to be able to say, like, we're seeing a lot of stuff happening and that make it look like it's going to look like this. And so we've seen that like these kinds of therapies employed at these times will be beneficial. And then it would change probably as time goes on, because a lot of these behaviors also they're pervasive, but then they can also be exacerbated in certain contexts. So I think it's all, a small piece of a very large puzzle.
Yeah, and I think the nice thing about sensory deficits is that we can figure out ways to perhaps change the environment that you grow up in to perhaps limit some of the deficits or mitigate some of these. But trying to figure out, like, there's certainly vision. It's fairly straightforward to do this. But yeah, not easy to implement them.
Yeah, definitely. So
Vivian,
overall, good paper.
I have a question I
Mhm.
should have asked at the onset. These genes that we've been talking about, these ASD-associated, are they associated in humans or just in mouse models?
On humans.
So,
Yeah.
that must be difficult because there's such a broad spectrum, right? And hard to pin down genes.
Yeah. So there's the Simons Foundation has a database. It's probably a Simons Foundation Autism Research Initiative, Safari. And they have a database with genes that have been associated with ASD, and they do a nice job of coding the genes in a couple of different metrics. And one is essentially being like, how prevalent or like how many links have been found, and then how confident are we about the links. So I often find myself being like, I wonder if this gene is involved, and I go look it up there.
Right.
But then from you will find that there's a big connection, a lot of back and forth between that database and mouse models.
Actually, I don't know much about the GABA B3. Do you know what that looks like in humans? So,
Actually, I don't know, but we can
yeah, because
According
Maybe
to
most
not.
of these, so MECP2 and SHANK3 and even neuroligin, mutations in those genes cause really severe intellectual disability and autism. So it's not just like, Rett syndrome, the girls really regress to the point where they can't talk, and a lot of the SHANK3 mutations, they never develop language. So these are severe intellectual disability genes. I'm not sure about the GABA one though.
Yeah, I didn't.
According to Wikipedia, it accounts for 1 to 2% of diagnosed autism cases, and it presents with classic symptoms that are associated with the disorder. It's part of the locus that is involved in the Prader-Willi-Angelman syndrome, which is another kind of part of this spectrum of syndrome.
I see.
disorder.
So, but there it may not be as severe as in like it's not like it's an intellectual disability per se.
Yeah, yeah. Wikipedia is less useful for that.
Yeah.
Yeah,
I
it
have to
looks
dig
like
further in.
this was identified in autism in 1998 by Cook et al. And it's actually also associated with childhood absence epilepsy.
Yeah.
Oh, that's how you pronounce it.
Oh, interesting.
Asperger's,
Absence. Yeah, because it's absence, right? But I don't know. I don't know how Americans say it, but that's how I say it. Absence. Association between the GABAB3 gene and autism, including one that enriched for savant skills. So yeah, I mean, there's more to look up, and if people are interested.
Okay. I literally typed in GABAB3 autism into like Microsoft Bing, and the first thing that popped up was not significantly associated with autism.
Oh, well, I don't know who that, so it does say, so it has like, it says, so I mentioned that the Safari gene database has a way of like scoring the genes. And it looks, so for example, it says high confidence. There's a high confidence. That doesn't mean that like it's, so let's see, total reports is 52 and autism specific reports is 28. So that's why I think it's important to have multiple metrics. One of being like, well, how confident are we about the science being true and a strong link, and what was the sample size, et cetera.
Well, I think that the authors of this paper should have delineated that a little bit more carefully, because it's very different to sort of associate a gene with autism versus having mutations in a gene that absolutely cause a severe intellectual disability / autism spectrum disorder. So they're conflating these genes together and saying, okay, the mouse models may be similar, but the human condition, they're not. But yeah.
But they also looked at, we focused on this GABA subunit for the purpose of this talk, but they also looked at like the other one, like MECP2.
Right.
And then in the previous paper, they also looked at like SHANK3. So there are like a bunch of maybe convergent evidence suggesting at least tactile linking
Yeah.
tactile.
Yeah. And what? How do you?
and, like,
the other one was. Is it? I don't know how y'all say it, but
I-
RORB or
Yeah, that one I don't know
RORB.
much about either.
Yeah,
Yeah,
I've
that I
only
never
I
understood
have
about
heard of that one, but not actually. I think about it in the context of cortex patterning and like neuronal identity. So it's always interesting to see when these like very like foundational transcription factors involved in giving neurons their identity are then involved in. I'm sure that's expressed in many other places, but it's always interesting to see that.
Thank you, Vivian.
My pleasure.
That's TWiN 51. Show notes at microbe.tv/TWiN. If you have any questions or comments, TWiN at microbe.tv. If you like this sort of discussions that we do on all these science programs of Microbe TV, we'd love your financial support so they can continue. You can go to microbe.tv/contribute. There are a number of ways you can help us out. Jason Shepard is at the University of Utah. Jason Synaptic on X. Thank you, Jason.
Thanks, Vincent. Good job, Vivian. That was fun.
Thanks.
Vivian Marsden's at Tulane University. Thanks, Vivian.
Yeah, no problem.
And Tim Chung is at New York University. Thank you, Tim.
Thank you. Thanks, Vivian. That was a very fun paper to read.
Awesome.
Vincent Racaniello. You can find me at microbe.tv. And listening to This Week in Neuroscience. Thanks for joining us. We'll be back next month.